this particular chapter will be difficult for most of the people, and I will be telling you how to solve the questions from this particular chapter in a very effective way. So I'll quickly summarize in 1 minute what are the various concepts that we have to read in this particular chapter. And I don't go too much fast, at the same time I don't go too much slow, because you are not a kids to explain word by word, but I'll explain in a simple and very effective way all people can understand. So please be active and participate and learn the depth of the concept. So in this chapter, we have to majorly read the process of replication, the process of transcription, the process of translation, and most importantly, friends, the process of gene regulation. The process of gene regulation, where you have to read all these. You no need to revise. That's what I'm telling to all of you. No one is nothing to record this. We will provide everything in the website. That's what I request all of you. Please attend in the starting of the class. Okay, we will discuss about the recording everything in the ending of the class. Now please involve in the concept. So we have to read replication, transcription, translation, as well as the gene regulation in case of a prokaryotic organism, also in case of a eukaryotic organism also. Apart from this, what else we have to read? We should also read a separate concept called as DNA repair mechanisms. Apart from this, what should we read? When we are reading the transcription, we have to read the post-transcriptional processes also. At the same time, my friends, we should also read the post-translational processes also in a very brief and elaborative manner. So before they use it, they never use it to give the question from the gene regulation in eukaryotes more oftenly. And even this particular 2022 question paper, they given it 2 questions, surprisingly, not even a single question. That is even in the part C only from this particular gene regulation. Hence, I encourage every student not to skip even a small topic, even a small information in this particular unit 3. So friends, all of you listen. When you are reading CSIR exam, one point you remember, it is not at all remember how much, I don't mind saying this line, it is not at all important how much time you read. It doesn't matter. It's the only matter how much time you, how much information you remember. More information you remember, more chances are there you can answer that question in a very effective way. Okay, now let's start it. So first we will go with the replication. So how to read the process of replication? So we should not directly read the process of replication. That is absolutely wrong strategy. When we are reading the replication concept, first we have to read the rules of replication. Then, my friends, we have to read the enzymes involved in the replication. And more important, friends, we have to read the processes. Whatever the PPT that I'm providing to you right now, it will be vary from unit to unit. If I'm teaching unit 1, if I'm teaching unit 4, if I'm teaching unit 5, if I'm teaching unit 7, if I'm teaching unit 10, my PPT will be 100% sufficient. But if I'm teaching unit 2, if I'm teaching unit 3, if I'm teaching unit 6, unit 12, unit 13, PPT will help you 80%, not 100% I'm talking about. So in the replication, we have to read rules, enzyme, then friends, ultimately followed by the processes. Now let's talk about this. And once the chapter get finished, immediately we'll solve all the practice questions from all the years, especially till 2016, not more than that, from the 2022. Okay, focus. You all know very well that DNA double helical structure was proposed by two intelligent scientists, James Watson and Francis Crick, in the year 1953. But friends, before the James Watson and Francis Crick, there is another scientist named as Paul. You know actually who is Paul? Paul is the teacher of this particular Watson and Crick only. You know, friends, what the Paul has been done? He has identified triple helical structure of DNA. What, friends? triple helical structure of the DNA. But you know, most of the scientists opposed this particular triple helical structure of the model. What is the reason? The reason, there is a no structural proof, there is a no experimental evidence to prove all the triple helical structure is the real model for the DNA. So that's why this model was completely opposed. Later, the students of Paul, that is Watson and Crick, they proposed a double helical structure with the help of what, friends? With the help of X-ray crystallographic structure taken by the Rosalind Wilkin majorly. Okay, later which she awarded the Nobel Prize. I mean to say later which she was shared the Nobel Prize along with the Watson and Crick. Friends, whatever we are going to read in the entire chapter of Unit 3 or Molecular Biology, entire information was summarized in this particular slide. Whatever we are going to read, focus everyone. So, what is DNA replication? First of all, why we have to read the replication? First, why we will only read replication? Then why we will come to transcription? Then why we will come to translation? Why we can't read the translation directly? Why we can't read the transcription directly? Why we are reading only replication? All of you pay attention, friends. Very important statement. We are all just because of our DNA. And if I ask you a simple question to all of you, what is the function of DNA? You all tell me generally DNA is the hereditary material. That is the first line comes in your mind. And what is the ultimate function of the DNA, friends? You all commonly know that the ultimate function of the DNA is what, friends? Synthesis of protein. Now you can ask me, sir, what is the major function of this particular protein? All of you listen. What not? Every function. 37.2 trillion cells present inside the inside the body. Every single cell, they will require protein to perform their function. Every function. Let's take a best example. Inside our body, hemoglobin is the one which will transport the oxygen. What is hemoglobin? Obviously protein. If I want to move my muscle, what I required? I required protein, actin and myosin. What are those? Of course, protein. If an antigen enter in my body, what my body starts synthesizing? Antibodies. What are antibodies? Of course, protein. So what not, my friends? Every single function performed by the living cell under the control of protein, that particular protein can be synthesized by this unique molecule, that is what? DNA. That's why we can say DNA is the most important, which plays a key role in the synthesis of protein. Now listen to me continuously for 15 minutes. Now the question arises here, sir, you said that DNA is the most important molecule involved in the synthesis of protein. That's absolutely fine. Then if DNA can synthesize the protein directly, why it is not involved in the synthesis of protein directly? Instead, why it is synthesizing RNA as an intermediate molecule instead of synthesizing the protein directly? That's not possible, my friends. Why? Why it is not possible? The reason, our DNA is a very highly stable molecule. What molecule? Very highly stable molecule. An example, just imagine, to protect its integrity, to protect its stability, the DNA always synthesize proteins with the help of RNA. An example, suppose example, if the RNA get damaged while synthesizing the protein, nothing gonna happen. We are having DNA just like that, it synthesizes the RNA. Then once again, we can restore that particular protein. But you only try to understand, friends, if the DNA get damaged, if we don't repair that, the same genetic information passes to the next generation, it become permanent, causes mutation, obviously leads to the cancer. So that's why, to protect its stability, to protect its integrity, DNA is going to synthesize an intermediate molecule, RNA. That intermediate molecule only, my friends, involved in the synthesis of protein, but not directly. Now, listen all of you. How does the particular DNA can synthesize the RNA? Before DNA synthesizing the RNA, DNA is undergoing the replication process. How can I define the word replication? We are all looking like our mother and father. Why? Because their DNA only, I mean to say, transferred from one generation to the another generation. So DNA replication is what? Duplication of the DNA, cloning of the DNA. So first DNA get duplicated or cloned itself, then DNA going to give rise to the RNA. Now all the students will get a doubt, sir, DNA is a very long molecule, but RNA is a very short molecule, which means that does the entire DNA molecule is involved in the synthesis of a protein? Never ever. It never involved. What is the answer for that? Why entire DNA is not involved? First of all, why the entire DNA has to be involved? It's not required, right? How much amount of the information is required to synthesize a specific protein? Only that much amount of the information only has to move from the DNA. It is going to be converted into RNA. An example I'm talking, I need only this much segment of the DNA. That's all. This amount of information is sufficient to give rise to this particular protein. Now, only this much amount of the DNA, what you can call it? A segment of the DNA, a small part of the DNA going to give rise to what, my friends? RNA. So, DNA mediated RNA synthesis. Once again, everyone listen my statements. DNA mediated RNA synthesis. What do you call it as? Transcription. DNA mediated RNA synthesis. Now finally RNA got synthesized. There is no doubt about it. Now RNA function is what we called mRNA, messenger RNA. It is bringing what the name itself suggesting, na friends. It is bringing the information. It is bringing the information from where? It is bringing the information from the DNA. Which information it is bringing? The information which is going to be translated into the protein. Only that much information brings the RNA, not the entire information present on the DNA. Now this mRNA combined with rRNA, combined with the tRNA, that we will read in the translation, they finally give rise to the protein. That is how protein can be synthesized. Now you can also say like this, my friends, DNA mediated RNA and protein synthesis. Of course, you can call it. Why? Because all of you listen, friends, DNA is like a hard disk which can store the entire information. So DNA only will guide RNA synthesis. DNA only will guide protein synthesis with the help of RNA. Do not forget that. Now, we all people know one thing commonly. 90% of all the organisms having DNA as their genetic material, we know that. So, the DNA is going to give rise to the RNA. We also know that RNA giving rise to the protein. But in certain cases, especially in the viruses, what is the specialty of viruses? They are having RNA as their genetic material. So friends, during that time, what is happening? RNA with the help of a unique enzyme. What do you call it, that particular unique enzyme? We will be calling it as a reverse transcriptase enzyme. That reverse transcriptase enzyme, from the RNA, it is synthesizing the DNA. All of you listen the statement very carefully. The synthesis of RNA from the DNA, what do you call? Transcription. The synthesis of DNA from the RNA, what do you call it as? Reverse transcription. So finally, we have developed a mechanism. We understood a mechanism that RNA can also give rise to the DNA. Friends, listen to me. Till today, the research is in progress. Inside our cell, really, is there any kind of a mechanism is existing that protein can give rise to RNA and protein can give rise to the DNA? If it's really comes into the picture, it's really a remarkable achievement in the biological field, but still the research is in progress. Really that kind of mechanism is existing in the cell or not, but really we don't know. These things every student can remember. So what you people remember has to remember for the part B as well as the one of the statements in the part C is their location. Remembering the location is extremely important, my friends. So please never ever forget the locations. Now, all of you listen. Okay. My voice is breaking. Am I audible to everyone else? Just type in the chat box. Suraj, I request you, please refresh. I am audible. Please refresh. Please refresh. Okay. That's great. Great, great, great. Okay, friends, please continue. Yeah, so you have to remember one very important point. What is that, friends? Where this particular syntheses are happening, that is very, very, very important. Everyone pay attention. Imagine, friends, this is a prokaryotic cell. Just imagine. And this is a eukaryotic cell. All of you friends pay attention. Now you know, right? Eukaryotic cell having a very well developed nucleus. Most of the students will forget this basics also. So pay attention. In case of a DNA, prokaryotes, what is happening, my friends? The DNA is going to be duplicated. Where? In the cytosol. From the DNA, who is coming? RNA is coming. So the process of transcription also takes place where in the prokaryotes? In the cytosol only. There is no doubt about it. But my friends, in case of in case of translation, obviously the protein synthesis also takes place in the cytosol only. My simple point to all of you is that in case of a prokaryotic organism, friends, the process of replication, the process of transcription, the process of translation, every single process is exclusively taking place in the cytosol only. Most of the students will get confused that there is a post-transcriptional processes occur in the prokaryotic organism also, which is absolutely wrong statement, friends. In case of prokaryotes, post-transcriptional processes never occur. What is the reason? All of you pay attention. During the process of transcription, when a little amount of the RNA comes out, my friends, I'm not talking about entire RNA also. When a little amount of the RNA comes out, that is sufficient. Immediately, what is going to happen? The ribosome complex will come and associate, and they start synthesizing the protein. Which means, what is the statement I want you to know? In case of a prokaryotic organism, the process of transcription and the process of translation is highly coupled. What, friends? It's highly coupled in nature. Both occur simultaneously. But you look into we ourselves, which we are who are very complicated structures, complicated genome or a complex genome, where the DNA replication is happening, obviously in the nucleus only. From the in the nucleus, what will synthesize? RNA will synthesize. Now the question is very, very clear. Do you think once the RNA synthesized in the eukaryotic organism, do you think protein synthesis will start immediately? That's impossible. Why? What is the reason? The reason, whatever the mRNA synthesized in the eukaryotes, it is not functionally active. It is not biologically active. It is immature mRNA. So my friends, immature mRNA cannot synthesize the protein. So what has to be happen now? This particular immature mRNA has to undergo the processing. We're going to read it separately, like 5' capping, 3' polyadenylation, then friends, followed by the splicing. All together, what do you call post-transcriptional processes? Why do you call it as a post-transcriptional processes? Because it is happening after the transcription. It's not a before the transcription, right? That's why we can call post-transcriptional processes, where that particular post-transcriptional processes will take place. Of course, in the nucleus only. Do not forget that particular point, friends. One more important point. Once the processing is over, the RNA has to be edited. Editing also takes place in the nucleus only. Finally, through the nuclear pore, where the mRNA will come. mRNA enters into what, friends? mRNA enters into the cytosol. Why the mRNA is going to enter into the cytosol? Because, friends, whether it is a prokaryotic organism or eukaryotic organism, doesn't matter. The process of translation or the process of protein synthesis has to happen in the cytosol only. That is the unique statement, friends. Never forget. Whether it's a prokaryotes or eukaryote, translation has to occur in here. And one more important point. See, friends, okay, finally the process of translation occurred. Done. We produced the protein also. There is a no doubt about it. And friends, that particular protein also not biologically active. You know, right? Let's take an example. When the insulin is synthesized, you called it as a pro-insulin. Why do you call it as a pro-insulin? Because it is not active, it's not matured. So same way, whatever the protein is synthesized, friends, protein is not active protein. So protein has to be modified. If the protein has to be modified, there should be a certain modification has to be occurred. Where that modifications are occurring? Of course, in the cytosol only. Whether it is a prokaryotic organism, whether it is a eukaryotic organism, doesn't matter. The process of post translational modification. Now you can ask me, sir, why do you call it as a post translational? Because the modification is happening after the translation. That is the reason. Now all of you listen, I'll summarize everything for all of you. I'm sorry. Focus, friends. In case of prokaryotes, listen, my friends, every one of you. In case of a prokaryotes, replication, transcription, translation, and my friends, post-translation, where all are taking place, all are taking place in the cytoplasm only. No doubt about that. Now let's look into the eukaryotic organism. What is you have to remember? In case of eukaryotes, all of you understand, replication, transcription, everyone, friends, I'll ask in the tomorrow class once again, and post transcription and RNA editing, where all these will happen, my friends, all these will happen in the nucleus only, in the nucleus only. But what about the translation and post translation? In the cytosol. I can tell you in a simple word. In case of a eukaryotic organism, friends, translation and the post translation, I am repeating, dear, listen. That's what I'm repeating for everyone. In case of eukaryotes, replication, transcription, post transcription, RNA editing, everything takes place in the nucleus. Whereas translation, post translation happening in the cytosol. But if you look into the prokaryotes, whether it is a replication, transcription, translation, post translation, exclusively taking place in the cytoplasm. One more interesting point. In case of a prokaryotic organism, there is a no post-transcriptional process, or there is a no RNA processing. Okay. Now everyone say just quickly yes or no. Understand till here? Now you can all ask all your doubts. Quickly, friends, say yes or no. If you have any doubts, you can ask me. Everyone, friends, everyone. Okay. Others. Good. See, friends, I will monitor every single student. I will put my 100% effort to the students who are feeling low, who are not understanding. But you just to show the interest, I'll put my 100%, I'll put my heart and soul to give you the best rank. I'm telling you sincerely how much effort I put to get the All India rank 2. I will put that much effort for all of you to give a very best lectures, and I will give you very best content. By that, you can face the exam very confidently. Okay, pay attention. Okay, I don't expect anything except your passion and except your interest. I don't want anything at all. Okay, now Now, next one, let's try to understand. So now we have to start what, friends? The process of replication. Okay. Now we have to read the replication briefly and elaboratively. And I also told to you, right? When we are in is mature RNA synthesis in prokaryotes, of course, dear. That's a good doubt, Devesh. Obviously, the RNA is a matured only, because that's what I'm talking. I just said, right? The DNA, once it synthesizes the RNA, that RNA immediately associated with the ribosome. Hence, the information whichever is coming, obviously accurate information only. There is no doubt about that. Okay, so it is 100% mature RNA. Yes, Srivani, even for your question also same answer. You got that? Okay, now understand. Now, the next, let's start with the replication. What did I say to all of you? When we want to read the replication, we have to read first all the basic rules involved in the DNA replication. We have to read all the enzymology involved in the DNA replication. Then only we have to read the process. Without reading the rules and enzyme, most of the students, what they will do, they'll just keep the book and they'll keep on reading the process. That's a waste of process. You have to know from very basic to the advanced level about their enzymology. Then only you can understand it. Okay, so pay attention. Let's start with the first rules of the DNA replication. Everyone, friends, pay attention. How can I define the word DNA replication? But so once again, please listen to me for the period of 20 minutes. Later, I will solve all your doubts. So kindly don't interrupt me in the middle, please. How can I define the word DNA replication? Biologically. How can I define the word DNA replication? Chemically also. If you ask me, sir, define it biologically, what I can say to you, it is a reaction in which daughter DNAs are synthesized using the parental DNA as the template. What do you mean by that? This is one parent strand. This is another parent strand. Two parent strands are separated and the two parent strands are acting as a templates. So my friends, what's happening now? There is a synthesis of daughter strand. That's a simplified definition, nothing difficult. A reaction in which daughter DNAs are synthesized using the parental DNA as the template. That's all. Okay. Now, if you ask me, sir, define the same definition in terms of chemical. So undoubtedly, what I can say to all of you, I'm going to tell you in the upcoming slide, what does the DNA is made up of? When I use a word, DNA is synthesizing. What do you mean by that? How can you synthesize the DNA? Synthesizing the DNA means addition of one nucleotide with the another nucleotide. That is only we can call it DNA synthesis, which means our DNA is made up of nucleotides. Can you count how many nucleotides? You can't. We have DNA having billions and billions of nucleotide. An example, you take the DNA and you keep it on the floor. The DNA goes from planet Earth to the planet Mars, and planet Mars back from planet Mars to planet Earth 600 times, not 6 times. That much long molecule our DNA is. What does this long molecule of DNA is made up of? Billions and billions of nucleotide. So how can I define the word chemically? Joining of one nucleotide with the another nucleotide. That is only we can call daughter strand synthesis. In a simple way, my friends, DNMP, deoxy, I will tell you one by one elaborately, just pay attention. Deoxyribonucleotide monophosphate. N represents number. My friends, this is the unique statement. When one nucleotide is going to be associated with the another nucleotide, all of you remember, it always comes in the form of a 3-phosphate, not 1 or 2-phosphate. One nucleotide, when it is combining with the another nucleotide, it will always come with the 3-phosphate. You will understand in the next slide. Here, you already read in the +1 level. N represents what? Number. So one nucleotide associated with the another nucleotide, it will become N plus 1. Out of 3 phosphates, how many phosphates are associated? Just 1 phosphate. What about the remaining two phosphates? My friends, remaining two phosphates are going to be released in the form of a PPI. That's a simple step. Now, I was repeatedly saying to all of you the same thing. Let me explain you elaboratively. I was repeatedly saying to all of you that DNA is made up of nucleotide. What is that particular nucleotide? What does the nucleotide is actually composed of? The nucleotide is made up of three individual parts. You should not say that, sir, does the nucleotides are made up of three together? That is wrong statement, friends. That never happens. DNA always, sorry, nucleotide always present individually. Later, with the help of certain links, all three come close together. Do not forget that point. Okay, what are the three individual parts? We can call the sugar, we can call it phosphate, we can call it bases. These three are the individual parts. These three individual parts will come close together and they can form one single nucleotide. That is one nucleotide. Now that one nucleotide associated with billions of nucleotide, finally they form DNA. Then imagine how complex molecule our DNA is. What does it made up of? It is made up of deoxyribose. You know, right? This is the sugar. What sugar we called it as? We called it as, my friends, listen to me, pentose sugar. Why did I call it as a pentose sugar? Having 5 carbons, 1, 2, 3, 4, 5 carbons, I called it as a pentose. But you only try to understand anyway. I will be explaining in the carbohydrate chemistry. Out of 5 carbons, how many carbons present in the ring? 1, 2, 3, 4. You can see, my friends, fifth carbon is not present in the ring light, right? It is like this. You called arm position. What do you call arm position? So how many carbons? 1, 2, 3, 4 carbons with 1 oxygen. What do you call it as? You can call it as what we call furan ring. Since it's a sugar, you called furanose. So furanose is the ring. Since it contains 5 carbons, my friends, we called pentose. Now you all know very well, if you look into the second carbon, if the second carbon contain OH group, then I will be calling this as a ribose. So we called our RNA all the time ribonucleic acid. The reason behind that, it contains, what does it contain? It contains OH group. But if you look into the DNA, my friends, we will be calling DNA as the deoxyribonucleic acid. What's the reason? The simple reason behind that, my friends, all of you understand. D means no oxygen. Only hydrogen is present. That's why we called it as a deoxyribose sugar. That's a simple point. Now, what does it contain? Phosphate group. All of you has to listen to me carefully. We, every time we use it to say that the DNA is a negatively charged molecule. Why the DNA is the negatively charged molecule? Just because of the presence of a phosphate group. That's a simple point. Now, how many phosphates are there? Only one phosphate is present in the nucleotide. Not at all, friends. In the nucleotide, 3 phosphate. Monophosphate, diphosphate, triphosphate. We can also call it alpha phosphate, beta phosphate, gamma phosphate. Anything you can call it. So, phosphate. Okay, now that's fine. What else the DNA should consist of? My friends, the most important one, the DNA should consist of bases. You all know very well, we will read in the chemistry part. There are 2 type of bases are there. We can call purines and pyrimidines. Always remember, friends, purines are the bicyclic ring. You can see now, sometimes in the exams they will say like this. They will give like this. Bicycling. And pyrimidines are monocyclic, single ring like this. Now, friends, adenine always pair with the thymine with the 2 hydrogen bonds. Guanine pair with the cytosine with the 3 hydrogen bonds. Okay, that's we commonly know that. We have read right now base separately, phosphate group separately, sugar separately. But if all come close together, do you think nucleotide, sorry, if all arranged separately, do you think nucleotide can form? Nucleotide never form. Okay, now all of you has to listen to me very carefully, friends. How These three come close together. All of you pay attention. This is the sugar, right? The first carbon of the sugar, which carbon, my friends? The first carbon of the sugar associated with the nitrogenous base by a bond. What do you call that particular bond? We will be calling it as a N-glycosidic bond. 90% of the students will understand this correctly, but some of the students will confuse here. Now, first carbon of the sugar associated with the base, no doubt about it. Now, all of you listen. Fifth carbon of the sugar. This is also unique, my friends. Fifth carbon of the sugar connected to the phosphate group. 90% of the students will say that this is the phosphodiester bond. That is wrong statement. This is not phosphodiester. This is called ester bond. Very widely, we will be calling this as phosphoanhydride bond. What we will be calling this, friends? Phosphoanhydride bond. Don't confuse, my friends. So the bond which connect the first carbon to the sugar, base, we call the N-glycosidic bond. Fifth carbon of the sugar connected to the phosphate group via ester or phosphoanhydride bond. You understand that clearly. Now you can ask me, sir, then what is the phosphodiester bond that we use it to read? All of you pay attention. Phosphodiester bond is the type of a bond. All of you listen. Phosphodiester bond is a type of a bond which is formed between two nucleotides. That's the reason we called phosphodiester bond is the backbone of the DNA. Why we call backbone? Which is supporting the two nucleotides. That's the reason I have given the name backbone. Now all of you pay attention, my friends. You look into this. This is one nucleotide. So I am drawing another nucleotide here. All of you listen. So another sugar I am drawing. The sugar associated with what? Bases. Whether it is a purine or pyrimidine doesn't matter. It also contains phosphate groups. How many phosphate group? 3 phosphate. Now every one of you pay attention very clearly, my friends. This is the OH group, right? Oxygen having what group? Oxygen having lone pair of electrons. Now that lone pair of electrons of oxygen, what they will act as? They can act as a nucleophile. So what is happening now? This oxygen nucleophilically, listen my statement. This oxygen nucleophilically come and cut one phosphate. How many phosphates? Only one phosphate. Then how many phosphates are going to be released, my friends? 2 phosphates are releasing in the form of a PPI. Now you can ask me a very logical question. Sir, here OH group cut the phosphate, 2 phosphates are released. Now the question is here, how does the OH and phosphate associated with each other very tightly? The answer is here. What is phosphate? Phosphate means energy rich molecule. Yes or no? Phosphate group is what? Energy. Now, whenever OH group, listen my statements carefully. Whenever OH group cut the phosphate, whenever it cut the phosphate, it generate the energy. That energy, I'm so sorry, it never generate, it lost the energy. That energy can be used to join the OH with the phosphate. So my friends, the bond which is formed between the OH group of one nucleotide with the phosphate group of the another nucleotide, we will be calling it as a phosphodiester bond, which we generally called, my friends, backbone. Simple. So this is one nucleotide, whatever we have drawn. Like that, billions of nucleotides come close together. Ultimately, they are going to form the DNA, which is a very, very complex molecule. So now let's start. We understand the definition of the DNA replication. We understand that DNA is made up of nucleotides. We also understand what is the structure of the DNA. Now what we're gonna read? We are going to read the major rules of the DNA replication. Now you can ask me, sir, is there any necessity to read every single rule of the DNA replication? I can say 100%. If you don't read the rules of replication, you can't understand the mechanism. At the same time, if you can't understand the enzyme, you can't understand the mechanism. So it's necessary to read the rules and enzyme, then the process will become crystal clear to all of us. Now listen, what are the various rules? Semi-conservative starts at the origin. I will explain one by one rule very elaborately. Synthesis always in the 5' to 3'. It can be unidirectional or bidirectional. Semi-discontinuous, RNA primers are required, then followed by the high fidelity. Fidelity means, what did I say to you? Accuracy. The accuracy should not be 99.99%. The accuracy should be always 100%, because even though 0.1% of mistake is present, if the same mistake passes to the next generation, it becomes permanent, causes cancer. That's why the DNA replication should be 100% accurate, not 99.99%. Now the first one, semi-conservative model. We will discuss one by one elaborately. Friends, pay attention. What is this particular semi-conservative model actually? You all know, right? Who, listen to me, I will explain slowly. Who proposed the double stranded DNA? Watson and Crick. You know, friends, what they have done, both people, after discovering the DNA, their entire research they published in the world famous journal Nature in the year 1953. You know, as a last lines of their publication, they have written two beautiful lines. What they have written, two beautiful lines, you know, they have written that base pairing helps us to what we can say, that the base pairing help us to discover or to make a model for the DNA replication. What is base pairing? You know very well, adenine pair with thymine, guanine pair with cytosine. Listen everyone, this base pairing actually given a hint to the Watson and Crick that they have to propose a model for the DNA replication. By using that model only, DNA can undergo the process of duplication or the process of replication. What is this particular semi-conservative model? What the Watson and Crick said actually, they said during the DNA replication, two parent strands are separated and acting as a template strands, and to this who are attached, friends, sorry, to which daughter strands are going to be synthesized. This is the simple semi-conservative model. Who proposed this? Watson and Crick. But you know, friends, 95% of all the scientists during that time accepted this model since it is proposed by a Watson and Crick. But remaining 5% of the scientists opposed. What is the reason why they opposed? How can you prove? The DNA is using only semi-conservative model to show the process of replication. What's the proof? There is no experimental evidence. There is no structural proof to prove semi-conservative model is the idealized model for the DNA replication process. So my friends, you know what happened? By opposing the semi-conservative model, some other scientists proposed two other models. What are those models? One we called conservative model, one we called, friends, dispersive model. Later, the two genius scientists, Matthew Messelson and Franklin Stahl, what they have done? They done a beautiful experiment. We use it to say one of the most beautiful experiment which were happened so far in the field of biology. Classical experiment. They proved finally semi-conservative model is the correct model. Conservative and dispersive models are not the correct model. They proved it. How they proved, we will understand in the next class elaboratively. Okay, is that clear, friends? Everyone till this slide? Everyone in the chat box, give me very quickly yes or no. Okay, that's really great, friends. That's really great. Okay, you can all people now, you can all ask me doubts. Okay, great. That's amazing. Okay, so friends, see, tomorrow also we are having the same class. When the class is 8:30 p.m. So friends, you are all able to see the website right now. Okay, now what you have to do, once the class is over, in the Google, in the toolbar, you just type sciencefestival.in. Once you type that, the website will come. Okay. Once the website will come, now on the website, you are able to see, right, free download content. Once you click on the free download content, here you are able to see three options: PPT 2022, recorded lecture June 2022, practiced test series. Okay. So once you click on the PPT, so tomorrow it will come. So since we did not upload it, right? Once the lecture is over, within 24 hours, you will get the PPT here and you will get the recorded lecture also here. Once the chapter is over, you will get the practice test series here. Okay. So this is how you have to access this particular website. Some organism may also have exception in case of semi-conservative model. Of course, true, Pooja. Sharma, it's absolutely true, dear. When we are doing the tomorrow class, we will do this. I will explain from there itself. Okay. And Pooja, I told you, dear, just keep it in your mind. Unit 3, Unit 6, Unit 4, Unit 5, then followed by the Unit 12, then Unit 7, Unit 13. Like that. Like that, I'll teach 10 chapters. Okay, Aishwarya, I will make one more class on about that on the YouTube very soon. Okay. So friends, I already told you, one day one chapter, one day another chapter. I will do 2 chapters simultaneously. And when it comes to the syllabus, I'm telling you very seriously, I'll finish syllabus like a rocket. Okay. Finishing syllabus is not the matter. Okay. So learning concept with the very depth of the concept is very important. I want you to learn it. Okay. So please kindly don't take it easy that someone is giving something freely. So by that we can access whenever you want. That is wrong thing. Don't do that. Okay. See, I know the value of money. See, when why I'm telling all this is because there are many channels are there where the people can teach easily and freely. But why I am saying very especially about me is I will put very dedicated effort on every single individual. That's why I'm saying I am different. Okay. So don't take it easily. Be consistent to every single class. Trust my words. I will pay attention towards every single student in upcoming days and I'll help everyone. Okay. Till you crack the exam. Okay. So friends, that's all for now. We will see you in the tomorrow class sharp 8:30 p.m. Okay. And tomorrow class, I will give the Telegram link. Sorry. I will upload all these PPTs you can find over there. Okay. And one more thing in the tomorrow class, what I'm saying something tomorrow is Unit 6 plant physiology. So listen, please keep on reading whatever I am discussing in the classroom. Once the chapter get over, we have to solve many questions, unlimited questions will solve. So make sure that you will understand and you will become okay. I know tomorrow is a Sunday, but since we have started class today itself, so let me take the class tomorrow also. I request all of you to attend. Okay, please attend. Don't think that it's a Sunday because we started today itself, right? So we will continue in the tomorrow also. Okay. Okay, friends. Thank you so much. Good night to all of you. Bye-bye. I'll see you in the tomorrow class. Bye-bye. Thank you all. Good night.
Thank you, sir.
Good night.
Thank you, sir.
Thank you. Good night. Sunday, there is a class. Okay.
Thank you, sir.
Thank you. Thank you all. Good night.